inflammation. There are three phases of acute inflammation. Fluid phase, it means the fluid is building up. Neutrophil phase, it means there is a rise in neutrophil, which is generally peaks around 24 hours. Macrophage phase is this peak in macrophage, which happens around 2 to 3 days after the peak of inflammation, right? So the peak of inflammation, acute inflammation is not defined by time because it can happen for days or even for weeks. It is defined by whether the neutrophils are the primary player or not. If pus is present, what is pus, by the way? Pus is just neutrophils in a fluid state. That's it. Dead neutrophils in a fluid state. That is called pus. If pus is there, it means it's a still neutrophilic attack going on, right? If you're not able to make pus, that means there is something wrong with your acute inflammation, because acute inflammation usually has pus in it, right? So pus is present, it's still acute. Okay, what are the steps of neutrophil exit? What is neutrophil exit, by the way? Neutrophil exit is just that this is blood vessel, right? Neutrophils are heavier molecules. They are really big in structure, and what happens is that in a flow of blood, they are usually in the middle of the bloodstream, right? This is one blood arterial, right? Imagine this is one blood arterial, and what happens that the blood is going through like this, right? It's going through like this, and in the middle, the neutrophils usually travel in the middle, so that the blood is not turbulent, right? So the blood is not damaging the vessel, the endothelial vessel. So that's why we need heavier particles to be in the middle, right? So what happens that the heavier particles, this neutrophil, they jump from the middle to the periphery so that they can go to this extracellular space, and then they can go to the intracellular space of any cell or any structure, right? So this jumping is called inflammation, right? When this neutrophils and fluids and protein, they leak from the capillary, from the arteries into the extracellular space, then to cell, it's called inflammation. What are the steps of neutrophil exit? What protein are the key in change in steps? What are the key proteins, right? So there are 5 steps. Step number 1, margination. What is margination? Well, margination, as you can understand, margins means this is a margin, right? Margination means usually a heavy particle in the blood are in the center of the vessel lumen, okay? When the blood vessel dilate, it means via smooth muscles. When these dilate, heavy particles come to the edge of the vessel. This is called margination. It's the first step in the neutrophil arrival, right? The heavy particle called neutrophil, they marginate themselves from the other part of the blood and they come on the periphery. Rolling. They are rolling. It means they are literally roll like this. They roll. So they're rolling. What's the rolling? Endothelial cells, right? The endothelial cells of the capillary. The endothelial cells show proteins such called as selectins, right? Which act as speed bump, right? So that neutrophil can roll and slow down. So this is one, wait, let me make it again. This is one endothelial. This is my endothelial lining. What happens that there will be upregulation of a protein called selectin, right? It selects these neutrophil, and there will be like these speed bumps. What happens is that these neutrophils, they will hit the speed bump and then get slow, because we need to pick them up. We need to, adhere them. What happens after this adhesion? What is adhesion? Just glue work, right? So adhesion, cellular adhesion molecule called CAM. Cellular adhesion molecule called CAM on endothelial binds to integrins on neutrophil, resulting in adhesion. So what happens is that your neutrophil produces something called as integrin proteins. These are integrins, right? And the endothelial lining here has something called as selectins. And to stop the neutrophil from leaving this whole environment, we need something called as CAM. This CAM actually holds selectin and integrins together, and so that this neutrophil can, jump. Okay, so adhesion. Cellular adhesion molecule called CAM on endothelial binds to integrins on the neutrophil, resulting in adhesion, right? Adhesion, what is adhesion? Adhesion is when one substance is attached to another substance. That's adhesion. Transmigration and chemotaxis. Well, first let's talk about transmigration. Transmigration happens across the cell wall. This is the cell wall, or post capillary venules. Okay, so when the jumping happens, it's called transmigration. Okay, across the cell wall. And chemotaxis is the movement to infection, after transmigration. So let's talk about it again. This is my blood vessel. There is my selectins. There is my integrins. This is my neutrophils, right? This is the CAM. When the neutrophils jump across the vessel wall into the extracellular space, we call it transmigration. And when from the extracellular space this goes to the infected site or the preferred site or the target site, we call this chemotaxis. Okay, the attraction of chemotaxis, right? Now chemotaxis, as you can remember, what are the four important things that perform chemotaxis? Attract, it's LT leukotriene B4, C5a, interleukin 8. Interleukin 8 was very important, and bacterial products. So these were the four things that perform chemotaxis, right? And it's done by cyclooxygenase pathway and 5-lipo. Do you remember, guys? It's about 5-lipoxygenase and cyclooxygenase pathway, right? Okay. So margination happened. So transmigration happened, okay, across the vessel wall of post capillaries, right? Why it happens at post capillary? Why not at arteries? Right? Why not at arteries? Why just happens it at venules? Well, not at arteries, because arteries are first of all very thick. They're hard to jump. Second, arteries have something called as tunica media, right? They are very thick. It's very fast blood flow, tight endotheliums, less fenestrations, and fewer cams are present at these arteries. Why it happens at venules? Because venules are minimal smooth muscle, right? You cannot jump smooth muscles, right? It's hard to jump via smooth muscle. You need something called as pericytes, and when pericytes contract, the gap between them, it increases, and then we can jump, right? Less thick walls, right? Slow blood pressure of the venules, right? More presence of CAM, the adhesion molecules, and pericytes contract, gap between them increases, right? So that's why it happens at venules. That's a very cool piece of information. Other thing, chemotaxis is the movement of infections to the infection site after transmigration. Neutrophils are attracted to bacterial products IL-8, C5a, and LTB4. LTB4, okay, very important. Okay, the last part is phagocytosis. Phagocytosis is just when this neutrophil, phagocytosis means this particle goes inside and is phagocytes, like it eats, it engulfs, right? So phagocytosis enhanced by opsonins, right? With opsonins, IgG and C3b. C3b, always remember, is the wife of neutrophil. It indicates what to eat, right? So opsonins, IgG, and C3b. So these are the 5 steps of neutrophil exit. This is very important. Let's talk about what induces P-selectins. What is P-selectin and what is E-selectins? What is it? Well, first of all, they are selectin molecules. It means they are expressed by endothelial cells of the blood of the vessel wall, right? So P-selectin, it means it releases induced by histamine. Now we remember that histamine is released by mast cells, and mast cells are, what are mast cells? Do we remember? Mast cells, what are the 3 ways to activate mast cell? Mast cell can be activated by tissue trauma, can be activated by complement protein, can be activated by cross-linking of IgE. Okay, so mast cells release histamine, and then this histamine releases something called as P-selectins. Now these P-selectins are released by Weibel pallid bodies in the endothelial cells. Another stuff made by Weibel pallid body is von Willebrand factor, which is very important for coagulation. What is coagulation? Insoluble fibrinogen convert into okay, I'm saying it again opposite. Soluble fibrinogen converts into insoluble fibrins. That's coagulation in short. So von Willebrand factor is really important in coagulation, and this is the same machinery that produces P-selectin, right? Another thing is called E-selectin. E-selectin is induced by TNF, tumor necrotic factor, and IL-1. Okay. So Selectin can be of two types, P and E, right? P is from histamine, and E is from tumor necrotic factor and IL-1, interleukin 1. IL-1 is released by oxygenase and 5L. Do you remember, guys? Oxygenase. And I'm talking about this pathway. Oxygenase means prostaglandins, and 5 lipooxygenase. Where is L-lipo? L stands for L stands for leukotrienes. Okay. What proteins on neutrophil does the selectin bind to? Right? Selectin is produced by endothelial, right? And endothelial selectin has to bind to neutrophil. How will it bind? It will bind. What protein on neutrophil does selectin bind to? It binds to silyl Lewis X. Okay, it binds to this protein. What upregulates cellular adhesion molecule? How CAM goes up? Well, again, this part, E-selectin. So TNF is tumor necrotic factor. TNF and IL-1, they produce E-selectin and they produce CAM. Okay, they produce E-selectin and they produce CAM. Same as fever as macrophage. Okay, same as fever and macrophage, because TNF and IL-1 also gives you fever, right? What are the major innate immunity cytokines? Innate, it's not humoral, it's innate. It's the first one, the less specific one. Same answer. IL-1, TNF. Okay, IL-1, TNF, and IL-1 is from what? COX pathway, cyclooxygenase pathway. You can stop it by giving paracetamol. It stops the COX. Once the COX is stopped, there will be no IL-1 going to perivascular space in hypothalamus, right? Once you stop that, you will not have fever. What upregulates integrins? First of all, where are integrins found? I told you integrins are found on this neutrophil. Okay, so you need integrins, you need selectins, you need something called as Lewis X. So what upregulates integrins? C5a, the complement factor 5a. Do you remember, guys, what was C5a? C5a, LTB4. Do you remember? These are the 4 things that cause attraction. LTB4, leukotriene B4, C5a, the complement part, interleukin from cyclooxygenase pathway, bacterial products. Again, we need, these are very important. Just print this in your memory. LTB4, C5a, IL-8, and bacterial products. Okay. Let's talk about a little bit about this acute inflammation more. What is leukocyte adhesion deficiency? If these leukocytes cannot adhere to the endothelial lining, do you think inflammation will happen? Of course, it will not happen. So it's an autosomal recessive. It's recessive means it's not dominant both alleles of the genes. It's a biallelic mutation. You need damage at both alleles of the genes to get this mutation, and it's the autosomal recessive mutation of integrins. Integrins means there is problems in neutrophils, and once there is, so why it happens? It happens because CD18 subunit of this CH is messed up. Okay, so CD18 is messed up, mutation at this point, CD18. Okay, what are the signs and symptoms of leukocyte adhesion deficiency, LAD? If you have LAD, what will happen? There's delayed separation of umbilical cord. What is happening here? What is happening here is that after the childbirth, the umbilical cord shuts down, right? You cut the umbilical cord, the whole system breaks down. Due to no blood supply, it undergoes necrosis. What goes under necrosis? The umbilical cord goes necrosis. Active inflammation ensues that. Okay, what happens? The necrosis happens because of the active inflammation. The neutrophils come and they start to eat up that umbilical cord, and soon the baby has no umbilical cord connected to its navel, right? So if there is no inflammation, the umbilical cord will, there will be a delayed separation of umbilical cord from the baby. So neutrophils won't come much because adhesion process is very bad. This is point one. Point number 2 is increased circulating neutrophils. What will happen that the adhesion is not happening? The migration will not happen, right? The migration needs integrins and selectins and Louis X, right? And we need CAM for the adhesion to happen. If adhesion is not happening, the serum neutrophils will keep on increasing because the factory that makes the neutrophil is all right. The problem is with the adhesion part. Okay, so increase circulating neutrophils, right? Normally half of the neutrophils are circulating because they need to be there if the inflammation keeps on going on, if the show goes down, right? So half of the neutrophils are circulating and remaining half are stuck on the endothelial of the lung. Okay, due to loss of adhesion, the circulating neutrophils will increase because these are not getting stuck to the endothelial lining. Recurrent bacterial inflammation but lack pus. Again, I told you, if you lack pus, there is some problem with your acute inflammation, right? So acute inflammation, acute bacterial inflammation, but no pus. The pus is dead neutrophils in a fluid. If neutrophils can't get into the tissue, how will the pus begin to form, right? Pus is made up of dead neutrophils plus fluid, right? So if there is no dead neutrophils in the cell, there will be no pus. And no pus equals to, you have something called this LAD. LAD equals to leukocyte adhesion deficiency, LAD. Okay, where do neutrophil exit the blood vessel? That is post capillary volumes. They cannot exit from arterial blood supply because arterial blood supply has a very fast blood supply. It cannot. They have to have a very slow blood supply, right? And a smooth muscle. They cannot jump smooth muscle. They must have pericytes, and pericytes are present at the post capillary venules where the blood pressure is less. How does the phagocytosis occur? Okay, neutrophil engulfs the stuff, right? Engulfs the foreign antigen, make phagosomes. What is phagosome, by the way? Phagosomes. Let's see, what is phagosome? Phagosome merges with lysosome to make phagolysosome, right? So once, okay, this is my neutrophil, okay, and this is the antigen. What happens is once it gets inside the neutrophil, once the phagocytosis happens, it will make how to say this, like a collide, not a collide, like a structure, and this structure we call it phagosome, right? If it's inside the neutrophil, we call it phagosome. Once it's inside, we call it phagosome plus what? Plus lysosome, right? Lysosome is for lysis, for killing. So lysosome plus phagosome gives us phagolysosome, right? So phagolysosome, right? Opsonins. What are opsonins? Opsonins are these mediators that increase phagocytosis. So C3b and IgG. Okay, IgG is for long term. IgM is for the acute inflammation. So C3b, where does C3b comes from? C3b comes from this C3. Do you remember, guys? C3 gives us C3a, C3b, right? Let's go back to this thing, convertase, right? C3 convertase converts C3 to C3a, C3b, right? Okay. What is Shidiak-Higashi syndrome? Cool name, by the way. It's an acute. It's autosomal recessive. It's a biallelic. You have to have hit on both alleles of your genes. Autosomal recessive protein. Trafficking defect. Trafficking defect means inside the cell, how these phagosomes travel. How will any substance travel? If a substance cannot travel inside, it means it will not happen, right? You need a good railway system inside a cell. Now this railway system is called trafficking system, right? So Shidiaki-Higashi syndrome, it's a trafficking defect. The microtubules. So what are microtubules? There are two type of things here: microtubules and microfilaments. Remember that microfilaments are smaller than microtubules, right? So these are big structural, railway path like this. Have you seen a railway path? You must have seen. So train travel on it, right? So microtubule railroad tracks in the cell are present for the trafficking of items inside. Once it's the trafficking damaged, you will have this syndrome called Higashi, this Shidiak-Higashi. What are the sign and symptom of Shidiak-Higashi syndrome? Very high yield. What happens is that it's the symptom based on impaired microtubule railroad, right? You guys have the substance inside the cell cannot travel. This microtubule railroad is the reason how phagosome travel with lysosomes. Okay, how the phagolysosome is formed via microtubule. If microtubule railroad is messed up, what will happen is that you will have something called as, first of all, immune findings, neutropenia, impaired cell division, low neutrophils, right? Bad railway road, bad nutrition, bad neutrophil development, giant granules, in leukocytes. Well, let's try to understand it. Newly made granules from Golgi body won't travel from one place to another, right? So what will happen is that there will be accumulation of a lot of granules here, a lot. And what will happen is that this will look like a giant granule. It will pile up. So giant granules. Increased pyogenic infection. Why? Because phagocytosis is not happening. It's not very effective. So phagogenic infection will happen. Phagosome merging with lysosome is impaired. So immune cells are less effective at killing the pathogens. Defective primary hemostasis. Defective, it means bad. Primary means primary. Hemostasis means the blood is stopped. Granules. So platelets have granules as well, right? So platelets, the granules in the platelets won't move well. It will result in primary hemostasis. Other thing, albinism. What is albinism? It's a melanocyte. Okay, let's talk about a melanocyte is the cell that produce melanin, and melanin gives you color. So melanocyte supplies melanin to about 25 different keratinocytes, right? These are 25 different stations of keratinocyte. Where are keratinocytes present? Keratinocytes are present on epithelial layer of your derma, right? So keratinocytes, there are 25 different stations. Once these melanocytes cannot work, the melanin won't travel, and so you will have, albinism, right? You will look white even though you are not white. As a railroad transport is defective, its melanin won't be transported to other keratinocytes. Okay. Peripheral neuropathy. What happens here that, for example, this is my spinal cord, this is my leg, right? This is my leg, this is my arm, this is my head, right? This is my another arm, this is my other leg. What happens that, for example, here's a receptor for my pain receptors, here's one axon, here's the cell body. What happens that this whole axon, these are long neurons, by the way. So if the trafficking is not happening well, the system will not work, right? Because the stuff is not traveling, the neurotransmitters are not traveling well. Stuff from the cell body of neuron won't be translated to the axon, ending well, right? So you do understand neuropathy, peripheral, because on the periphery you have long axons. That's the whole concept. So that was acute inflammation. Very small topic, very easy to understand. I hope you really understand it very well. Okay, have a nice day.